Yes.
Okay, all right. So I wasn't sure what to do, so I thought I would task myself with looking at a couple of recent developments in the HIV cure space, especially the gene therapy aspect of it, and try and give you an update. So I'm a pretty informal speaker, so feel free to interrupt me. Um, and I thought to sort of center us, first of all, I want to remind you of what I think of as like the two big problems that HIV presents to us. So number one, it just basically defeats our natural immune defenses. I think in the time of COVID and vaccines, we're all becoming, expert immunologists and understanding that our immune system should be able to wipe out a viral infection. And our immune system can't do that with HIV. And we're also failing in our task to even develop vaccines against HIV. So what's going on? So, firstly, it's a very smart virus. It carries genes that are, their role is to disable parts of our immune defenses. And then secondly, this virus mutates at really high frequency. Again, you've probably all been hearing the recent press around, shock, horror, coronavirus variants. The coronavirus is mutating a little tiny bit after 250 million people have had it. It's just, the coronavirus is a wimp compared to HIV, which even within one infected individual just produces so many mutations at such high frequency. And one of the consequences of that, of course, is that even if people make really good antibodies that should wipe out HIV, the virus can often just mutate around them. And then finally, and this is the thing that I'm going to stress, because one of the studies I want to highlight really addresses this, is that HIV, for whatever reason, decided that it was going to infect and kill one particular cell in the body, which is the CD4 T cell. And the CD4 T cell is just such an important, critical part of the immune system that the consequences of that are that many aspects of our immune defenses just fall apart in the presence of HIV. So that's just, I guess that's like a reminder. You guys should know all this by now. So I said this was problem one. So then problem two is this other trick that HIV has, that once it gets into a cell, it permanently integrates its genome into the DNA of that cell. So my little picture up at the top here I'm not very good at art. This is a virus entering a CD4 T cell, and this is its genome now becoming a permanent part of that cell, so that the virus is always present in that cell. And even worse, sometimes this integrated HIV genome can become latent. It switches off, probably not very frequently, but if you've got billions of these viruses in your body, you're going to have a high number of these latent viruses.
I'm on Zoom. Oh, okay. Do you need something?
No, it's okay. That's something.
Oh.
Just a reminder to everyone, please mute yourselves while the speakers are speaking. Thank you.
Okay, so this virus can become latent, and the end result of that is that it persists even under long-term use of antiretroviral drugs. And my little cartoon down at the bottom is just, I think you'll all be familiar with this. This is showing what happens to levels of HIV in the blood when somebody starts antiretroviral therapy. The levels are undetectable. But of course, if you stop the drugs, in most cases, the virus comes roaring back. And one of the things we want to do with an HIV cure is either completely get rid of the virus, or at the very least, enable it so that even if you stop taking antiretroviral therapy, you can keep your virus under control. All right. So then, continuing with a bit of background, let's do some immunology. So one of the things I think about, and I'm not an immunologist, so some of this goes over my head, but our immune system is really a team effort. So here I'm showing three of the key players: the CD4 T cell in green, the CD8 T cell in purple, and the B cell in blue. And they all do different things. So let's start with the B cell in blue. The B cell makes antibodies. Okay, they're really important in combating viral infections. The CD8 T cell these recognize infected cells or rogue cells, such as cancer cells, and they actually kill those cells. So the idea is that the combination of antibodies mopping up free virus and then the CD8 T cells coming along and killing any viral infected cells should enable you to control a viral infection, including HIV. And then the CD4 T cell has this kind of organizing role. We say it provides help, and you may also have heard of them called helper T cells or CD4 helper T cells. And they provide help to both the B cells and the CD8 T cells, which can't do their jobs very well in the absence of this help. And so this leads us to the sort of dilemma of HIV infection. So if HIV infects a CD4 positive T cell, what you're going to get is, first of all, the CD4 T cell is going to be making new HIV viruses. And also on its surface, you'll see some of this is the envelope protein or GP120 on the outside of viruses. You'll see some of that decorated on the surface of an infected cell. And so, along comes our B cells, and they make the antibodies, which are secreted, and they're going to neutralize the viruses. And then also along comes our CD8 T cells, and they have something on their surface called a T cell receptor that's a little bit like an antibody, if you like, but it allows the CD8 T cell to recognize this as an infected cell and to kill it. And again, we have the CD4 T cells also providing help to allow the B cells and CD8 cells to do their job. But then the problem is, of course, that if those CD4 T cells get infected by HIV and get, wiped out by HIV, then we lose this balance, and our CD8 T cells and our B cells are no longer able to control HIV, and so the virus can now continue sort of unchecked in our bodies. So that's the dilemma and the challenge that's presented by the fact that HIV infects CD4 T cells. It really does a number on our team immune system. Okay, so gene and cell therapies, there's been a lot of advances, but the approaches are still in three kind of buckets. First of all, the idea that you could make the target CD4 T cells somehow resistant to HIV and protect them from being infected. And John is going to talk about that after me, in particular, work he's been doing to disrupt the CCR5 co-receptor. You can also think about ways to enhance the immune responses, being better than nature, if you like, by engineering B cells or CD8 T cells. And I'll show you one example of that. And then finally, there are ideas around the idea of purging the reservoir of these latent HIV proviruses. And I'll show you a recent development there. Okay, so I'm going to go over a couple of studies that have been published recently. So first of all, the idea of making better CD8 T cells. So if you remember, this is how a CD8 T cell recognizes and then kills an HIV infected cell. It's through this molecule on its surface called the T cell receptor. And T cell receptors, you can build artificial T cell receptors. Sometimes we call these CARs, stands for chimeric antigen receptor, but you can just say CAR. And they are engineered to be really good at recognizing the parts of HIV that are expressed on the surface of the cell, and therefore making an even better CD8 T cell than perhaps nature could make. So one study I wanted to draw your attention to is from a company called Lentigen. And the reason I've highlighted this is because just in the last couple of months, they've started a clinical trial. And these numbers up here are if you go to clinicaltrial.gov, you can find the numbers. And they're going to be recruiting through Steve Deeks at UCSF. And the idea here is that T cells will be taken out of an individual and then engineered in the lab with something that Lentigen called their GP120 duo CAR, and also expand the cells and then reinfuse them. So what is this duo CAR? Well, basically it's the idea that they are going to take a CD8 T cell and give it two different engineered T cell receptors. they're kind of shown down here. This is a figure I took from the paper that was in Science Translational Medicine. And these duo CARs recognize what's good about the combination is that, is that they recognize two different parts of the HIV GP120. This is a CD4 binding domain, and this is, I can't remember. I think it recognizes another part of GP120. But the idea is that by combining these together, you might be able to get an even better CD8 T cell. And certainly the data they had in vitro in their paper suggested that. So this is a clinical trial that's just started enrolling. The next trial I want to talk to you about, and I apologize, it's a very busy slide, but I'll walk you through it. This is from a company called American Gene Technologies. Again, only a couple of months ago, they started a clinical trial, which is going to be enrolling individuals in Washington, DC, through Georgetown University, I believe. And they are going after the idea, if you look over here at this cartoon, remember this idea that a CD4 T cell is not just the target cell for HIV, but it's also the cell that has to give help to make really good CD8 T cells. And so their idea, which I think is quite clever, is that they understand that these HIV-specific CD4 T cells are needed for help. They can isolate these from the blood of an HIV-positive individual. They're present, but they're at very low frequency. So they'll typically take, for example, 1 million of these cells. Then, using their sort of technology, they can massively expand the numbers of these cells outside the body by stimulating them with peptides that are part of the HIV gag protein. And at the same time, they introduce a lentiviral vector that contains some genes that will make that CD4 T cell resistant to HIV infection. And then they take this kind of expanded, stimulated, and engineered mass of CD4 T cells and reinfuse them back into an individual. And, you know, they go from about 1 million at the start to 1 billion at the end. And the idea here is that they are expanding the quality of the CD4 T cell help and protecting those CD4 T cells from being infected themselves. And thereby, they should be magnifying the ability of the CD8 T cells to kill any HIV infected cells in the body. So it's kind of a cool approach that takes advantage of the team approach that the immune system uses to tackle HIV. Okay, and then this is just me sort of reminding you of this team approach. And so the lentigen strategy is making a better CD8 T cell by engineering 2 different TCRs. And the American Gene Technologies approach is making better CD4 T cell help by expanding and making these cells resistant. Okay, so then the last thing I wanted to touch on was to go back to this idea that HIV integrates into the DNA of a cell and can persist and form a so-called latent reservoir. And this is really a roadblock to people coming off antiretroviral therapy and not having viral rebound. So people are thinking about ways that we could purge this reservoir. And there's a group at a company called Excision Biotherapeutics who have a very ambitious project. And the idea here is that they are going to use CRISPR, which I always think of as just a pair of scissors, to cut up the integrated HIV virus in a way that will produce some quite large deletions. If you can imagine having a cut here and a cut here, and then the pieces in between are dropped out, you're basically removing most of the HIV virus from an infected cell. And the way they want to do that is they are packaging the CRISPR scissors into a vector called AAV9. And they have some proof of principle studies that they just published I think a couple of weeks ago, using rhesus macaques, where the rhesus macaques were injected with 10 to the 13 vectors carrying CRISPR-Cas9. This is a really huge number. I was trying to think, what does this number mean? And then I realized that 10 to the 13 is also approximately the number of cells in a body anyway. So they're kind of adding, enough vector to infect every cell in the monkey. And they had these monkeys on antiretroviral therapy, so no information about whether this allowed the monkeys to control their infection. They're infected with HIV. But what they did report in their paper is that they saw evidence of this cutting out of the virus. And over here on the right, I'm showing one figure from their paper where they went in and measured the amount of total HIV DNA in the monkeys before and after treatment, and they saw various levels of decrease. Now, this approach gets quite a lot of publicity. And I have to be honest with you, I don't think it's ready for prime time, and I have some real concerns about it. As a scientist, I'm always very concerned by anything that only has 3 monkeys and 1 control, and the control is also going down. So I have to say I'm not completely convinced by their data. And also just, I recognize this is just a proof of principle, but of course, this idea of mass delivery into an individual of AAV vectors carrying CRISPR-Cas9 raises lots of concerns about what those vectors are going to do, including insertional mutagenesis, potential carcinogenesis, and just you may have a massive immune response to the AAV vectors. So there's a lot of things. It's easy to criticize, but I give them credit for persevering with what I honestly thought would be a complete non-starter as an approach, and at least having some incremental progress to show that this may be feasible. Okay, so just to summarize, back to this overview slide of the different types of gene and cell therapies. Again, with the protection, I'm sure John's going to talk about this in more detail. With the attack, the enhancing the immune responses, I showed you the lentigen and also the American Gene Technologies approaches to improve CD8 T cells and improve CD4 T cell help of those CD8 T cells. And then finally, this idea of in vivo delivery of CRISPR to cut out the HIV. And I'll leave you with Timothy, because whenever I give a talk about HIV cure, especially this year, I'm always going to end thanking my friend Timothy. Okay, so I will stop sharing, and if we have time, happy to answer any clarifying questions, or we can just move on, Jeff, whichever you prefer.
Yeah, there's one question that might be good while John brings up his screen. The screen sharing is enabled, so go ahead and do that. Bob wanted to know if any of the recent advances in mRNA technology for the COVID vaccines have any application to HIV.
Oh, I think so. I think so, absolutely. I mean, the great thing about the RNA technology is you can really, it's so flexible. You can really change what you put in. So the idea that we could come up with some sort of immunization, for example, that would be a vaccination, and instead of just being one type of the HIV GP120 protein, you could put in the RNA code for 20 different types, and thereby potentially induce a complex mixture of antibodies that together might be able to attack the virus, or even train our own immune system to produce these sort of powerful, so-called broadly neutralizing antibodies. I really feel that a lot of the things that we've not been able to do in terms of vaccination, both to protect people and also therapeutically, are now kind of on the table, because the RNA technology just gives us a flexibility that we don't have when instead we are making, proteins that are going to be injected or viral vectors. So I'm excited to see what might happen there.
Great, thank you so much. I think that's a good segue into Dr. Zaia's talk. So take it away. Make sure you're unmuted.
Okay.
There you go.
Fine. Here, you put the screen up, you can't see the mute. Okay, well, so I'm going to continue this thread of discussion by splitting my talk into two areas where genetic therapy, I think, is playing a role. First is exactly what Paula mentioned, and that is use of immunotherapy. That is how to make better T cells, or at least make T cells function better in this area, because ultimately that is the missing piece. If you could do that, then you probably don't have to worry about all the other genetic manipulations that we're doing. And certainly the model, if the model of HIV is if the metaphor is leukemia, and you have to look at the cellular disease, let's say, and get to the last cell to evoke a cure, then we know that T cell therapy can do that. So CAR T cells are, I think, an interesting thing to talk about. I want to just talk about one area, and I think that the trials that Paula mentioned are really going to be very interesting. So I'm going to talk about our approach. I'm trying to figure out how to get this to go forward. Here we go. So of course, CAR T cells have been around for HIV for a long time, as you know. The first CAR T cell therapy that was really a systematic approach was in HIV over 20 years ago. And the interesting thing is, why didn't it work? Well, of course, that CAR T cell was the one that was made to have a CD4 on the outside of it. So it would go out and search for GP120. And that worked really well in vitro. In vivo, it didn't work. In those days, they didn't have the kind of co-receptors that we have now that will activate the cells and also make them last longer in vivo. And those cells also were not protected and could get infected with HIV. That is, they didn't have a genetic protection built into them. But interestingly enough, 10 years later, they still could find, Steve Deeks actually did a phase 2 trial. There were a lot of patients treated in those days. Tremendous amounts of money were spent 20 years ago on CAR T cell trials for HIV. And I suppose you guys know that pretty well. So, but even 10 years later, you could find these cells in the recipients of those treatments. And the question is, why is that? And one of the theories is that some of the cells that got taken out of the donors to be genetically manipulated and put back in were long-lived cells that probably recognized endogenous stimulants like CMV, cytomegalovirus. We don't know that for a fact, but it's possible. And that's one of the themes I want to follow through on. So CAR T cell therapy is exactly what Paula said. I don't think I'll spend any time on this slide. Take the cells out of the person, genetically modify them, put these chimeric antigen receptors on them, put them back in, and hope that they will then kill the HIV infected cell. And one of the problems, of course, is during CAR therapy, there is no HIV. Oops. Let's see what I'm doing here. There is no HIV stimulation. And a CAR T cell is hungry for antigen. If it doesn't see it, it dies. So that's one of the problems, is how do you handle the problem that CAR T cells need to see HIV when they go into the body? So the way we thought we would do it is to give them, again, it's very much like the duo CARs that Paula talked about, lentigens. Only here we're going to start with CMV infected cells and genetically manipulate a CMV. I don't mean a CMV infected cell, I mean a CMV reactive cell, a CMV specific T cell. And we know that HIV, that as persons living with HIV, have a lot of CMV reactive T cells. It's one of the favorite antigens to react to, you may have 1, 2, 3% of your total T cells are reactive to CMV. So these are the cells that you genetically manipulate to make to put on the surface these red. So they have the green CMV receptors, and then you put onto them the red HIV chimeric antigen receptor. So they have a natural T cell receptor and a chimeric antigen receptor. And so these, you can call these duo receptor cells too. And we can actually stimulate these cells with a CMV vaccine. And then you inject them back into the person and see what happens. So that's the theory. So that these cells will get help from the CMV reactive cells. They'll flourish. They won't die in the desert of a ART-treated a person. So the antiretroviral therapy can continue, and then you can take it away and then see if these cells modify the rebound. And this is actually what it looks like. This is the HIV infected cell with a GP120. So we use N6, which is a broadly neutralizing antibody. We took the SC receptor, made a linker, and then this is then injected, put into a transmembrane region and has the 4-1BB as the co-receptor. And it has the CMV part of it, and we can activate that with the CMV vaccine. This is the endogenous CMV T cell receptor. And we also put a marker on it. This is truncated EGFR, and it is something that an antibody, cetuximab, can be used to mark the cells, or you can actually use them to kill the cells if the cells got out of control. And so the whole treatment strategy looks like this. You take the HIV donor who's on ART, you get his peripheral blood mononuclear cells, you pulse them with CMV. They're going to start making interferon. You use interferon beads to pull out the cells, and now you have a bunch of purified CMV cells, and you then take some lentivirus and you modify them so they now have the chimeric antigen receptor on the outside. You can stimulate them with the vaccine, and voila, you have CAR T cells proliferating in the absence of HIV antigens. So we're actually putting together an IND because we've got a lot of data now that shows that we can actually do this. This just shows you a collection of CMV specific cells. And they're actually mostly CD8 cells. So we don't have to worry about them getting infected. So we don't have to protect them with some endogenous materials like others have had to do when you're using more CD4 cells. They tend to be some memory cells in there. They're not all memory cells, but some are central memory cells. Some of these called TEMRA cells, which are also a form of memory cell. So we think that they will last for a while. They actually don't, even though you expand them, there's very low expression of exhaustion markers. And that may be because of their CMV memory cells. CMV cells tend not to have a lot of exhaustion markers. So these are exhaustion markers, PD1, LAG3, TIM3. They actually can control CMV reactivation in vitro, and they can kill infected cells. So this just shows what I've already shown you before. And we're not growing the cells with antiretroviral agents like we would clinically for the clinical trial. We're just letting the virus come out of these cells in culture, and we're adding CAR T cells or not, and seeing what happens. And you actually get the CMV cells. The CMV cells can react to CMV antigens, and they can show reactivity by interferon to CMV. But they also control HIV. So this actually shows you the CAR T cells with CD19 CAR. So we can also make CMV CD19. Now this is a B cell, obviously a B cell reactive T cell, and it's used for leukemia. And we actually do have a clinical trial that we'll start here this year for the use of CMV specific anti-CD19 leukemia cells. That trial will begin, and that will also use CMV vaccine to induce killing and persistence. But the point I'm going to try to show here is that these cells do kill a cell that expresses GP120. This here is after 21 days in culture. We're looking at the percent of cells that P24, and in fact, the CAR T cells have basically reacted to the endogenously reacted cells and suppressed them, and the CD19 cells could not do that. So it does control HIV reactivation in vitro. This here is an attempt to show what happens in a mouse model in which you're infecting them with, it's the PBMC model in which you infect them with PBMC infected cells. You put in the CAR T cells, then you sacrifice them at day 55, and you look to see where the cells go. They actually go to a number of places. This is the mouse. They're in the bone marrow, and they're in the peripheral blood, and a small amount of them are in the spleen. And what kind of cells are there? It's almost all CD8 cells, again, because they came from an HIV positive donor. But there are a few CD4 cells. The vaccine increases the number of CAR T cells in HIV infected mice. So we had to show that the FDA wanted us to prove that the vaccine actually does something. So it does actually increase the number of cells that are in this mouse model. And then finally, does it control rebound post-ATI? So this model here is, again, your PBMC model. It goes into the mouse. You get CAR T cells on day 19, but you've actually started the mouse on oral antiretroviral therapy. So the T cells come in when there's no HIV or limited HIV. And this model does not eliminate circulating HIV. So there is a low amount of it. And then you give the vaccine to expand those cells, and then you take them off ART to see what happens. And this is day 28 while they're on ART. And you can see that the high dose of CAR T cells actually did suppress, even in addition to the ART, and the low dose did not do as well. But it was, and this is without any CAR T cells. But then if you look at the post-rebound, the high CAR T cells did control the rebound, whereas the low number of CAR T cells or no CAR T cells did not. So we're taking this to the FDA, hoping they will let us go forward. We have a trial that we plan to execute at the University of California, San Diego, with Davey Smith as the PI. So we're going to screen and enroll, and then CMV vaccinate the volunteer, HIV positive, collect their cells, manufacture them, put them back in, give them the vaccine, and then stop ART and follow them for 2 years. So that's that project, and this shows the characters. It's really the work of Julie Wang and Angelo Cardoso primarily, with John Burnett doing the animal work. And I'm showing our advisory group as well, statistically, and Karen has been very helpful to us on planning on how to evaluate the patients during this trial. This shows Davey Smith, and Elizabeth Buddy will be the PI at City of Hope. So that's our CAR T cell thing. So the question, the thing that Paula referred to was, what can you do with stem cells in terms of knocking out CCR5? And wouldn't that be a better way to get T cells out of the bone marrow than having to do all this other extraneous preparation of T cells that can be quite expensive? So we'll talk about how to do a bone marrow transplant with genetically modified cells. And it could be an allo transplant, of course, but that's difficult to do. Autologous transplantation is what we did. And so that's the donor is the self. The donor obviously is oneself. So we're going to collect the cells, put them through a selection system, pull out the CD34 cells, do the genetic editing using zinc finger nuclease by electroporation. This is an electroporator, and then transplant them back. And in fact, when we started this, no one had actually ever taken stem cells through the electrical, let's say, stimulation that is necessary to get these zinc finger nucleases in which will then knock out the gene of interest, and then get them to engraft. So that was really the question: can you actually do this? And this is from Paula. This is her famous slide that I'm sure she's only shown about 10,000 times on zinc finger nuclease. And this is the cutter, of course, and it leads to CCR5 disruption. And you can measure that in various ways. And this is, of course, the CCR5 gene. And that's the target right there, right at the beginning. So you knock out the whole gene if this works in a cell. Of course, you have to get 2 genes, and you have to knock out both the genes, or else you're going to have some CCR5 still expressed. So this is the trial that we began several years ago. And Paula did all the work to lead up to the IND that we did. And then Amrita Krishnan was the clinical PI, the transplanter here at City of Hope, who actually did all the transplants. And we worked with Sangamo. And it was their technology really that we were using. And CERM provided the money. And we found several sites where patients could be enrolled and then screened, consented, sent to City of Hope, treated, and then sent back to the site. So Tony Mills here in LA, Jay Lalazari, and Ron Mitsuyasu also here were the main people. There were a few other sites, but no one else contributed patients. These were the main patients, the main centers that contributed 9 target patients. 1 patient actually was found to be ineligible, so we ended up treating only 8. And this is the study summary, just to go through just very briefly. You had to have R5 virus. You could not have X4. That was the hardest thing to find. And then this was the product. It was a hematopoietic stem cell that we infused into the patients. It was done after busulfan conditioning. And it was a complicated thing to put together because people had to fly in from another site or drive in from LA, obviously. But a drive from LA to Duarte here is sometimes a long drive, certain times of the day. So they would come here, we would mobilize their cells, do the apheresis, collect the cells. We had to do 2 apheresis. You had to use plerixafor as well, not just GCSF, to get tremendous numbers of cells. And then you had to manufacture, and that took about 6 weeks. So they would go home, then they would come back for their busulfan conditioning, and they would stay here for about 3 weeks until the cells engrafted. There were 2 cohorts. One got low dose busulfan and one got higher dose busulfan. That's still only half the dose that would be used, let's say, in a sickle cell patient that's getting the same sort of gene therapy. So we didn't want to go very high in these otherwise very healthy people. And then once they reach certain level of engraftment and recovery, you could do an analytical treatment interruption, but you had to have CD4 cells return to at least about 400. Although we got away finally with 350 if you had 25% of them were CD4 cells. So that's the big picture. So this just shows how hard it was to actually find these patients. We screened 71 patients. We had 57 of them were screen failures. The reason they were screen failures for various medical reasons, CD4 count reasons. They had been, some of them were found to be just coming off another trial, which wasn't permitted. Some had a SNP that altered the CCR5. But by and large, it was the HIV tropism. 40% of the failures were due to the fact that they had X4 virus. Some withdrew the consent after they got here. They got here and they say, wait a minute, I'm going to have a bone marrow transplant. So that was the kind of thing that happened as well. But they all engrafted. So this just basically shows all the patients, and it shows in color either blue the white cells, green the absolute neutrophil count, red the platelets, and so they all engrafted. Now the X's are when we give GCSF, which we do to make sure that they do pretty well. That is, they get engrafted faster than on GCSF. At one point, we decided we're probably mobilizing cells that are already genetically disrupted. So let's not do that for the last 2 patients, which we didn't. So you can see they didn't recover as fast. And they actually did have a slightly better percent of disruption. So the research outcome, this just shows the CD4 counts. So the CD4 counts come back very slowly after an autologous transplant, much slower than we had hoped for. And that, I think, will ultimately be a problem. Can an HIV positive person who's an adult have the kind of thymic function that you need? You see a very robust recovery in some autologous transplants, but the older you get, the harder that is. And in an HIV positive person, that may never be correctable. Some recovered pretty well, but some have taken a long time to recover. It shows the percent CD34 cells. This shows the disruption. So we actually got, we got disruption. It wasn't the greatest disruption, but we were actually able to get in the range of about 1 to 5% disruption that was then lost over time. And with time, we have a subset that maintains their engraftment at about 1%. Still very low, certainly not enough to correct an ability or produce an ability to control HIV infection. And why is that? Why is that? Well, the zinc finger nuclease technology itself was not as good as it is today. Today, they can produce, 70 to 80%, even 90% disruption of a specific gene. In the days when we were doing this, we were lucky to get, 50%, and actually in the clinical trial, we got closer to 20 to 40%. So that in itself was a problem. But the use of busulfan as a conditioning regimen is let's call it a weak conditioning regimen. For sickle cell disease, you would have blasted the bone marrow and emptied it with busulfan, and then you get good engraftment. We couldn't do that, of course, in this setting. And until there is a really good way to do safe conditioning, let's say with antibody or something like that, which will be very specific and non-toxic, this technology is going to have a problem. But we looked at the conditioning regimen with busulfan. We looked at the efficiency of disruption of the gene. And we looked at the quality of the stems of the infused product to see what was going on. And in fact, we did achieve the busulfan levels that we aimed for, which is not that easy to do. You have to maintain, you have to do a test dose, and then you have to infuse multiple times based on modification of the dose, based on the pharmacokinetics. We got disruption. So this is the low dose and this is the high dose. So we got great variation between patients. But with the higher dose, and we did modify the method with the second cohort as well, we got fairly consistency and we got better disruption. But again, this is not 70, 80, 90% that you could have produced today with zinc finger technologies. What about the viability? One question is, are these cells, after they've been electroporated and frozen, and you saw them, are they really that viable? Yes, they are. We found that viability at the bedside was outstanding. We were really surprised at that. What about the stemness? Did you actually alter the stemness of these cells? Well, stemness is the ability to reconstitute, let's say, a mouse who's been irradiated. But in the human system, this is thought to be the lineage for really for the most important hematopoietic progenitors, let's say. So we could actually look at that in our product. And in fact, you could see that. So this is the primitive lineages that leads to stem cells. This is the most primitive here. So, and they are very few. They're usually less than 5%, or much less than that, often 1%. We actually saw some pretty good percentages of the most primitive of the lineages, and these other lineages, which are still CD34 positive, are also there. So we felt that we actually did get really good stem cells into these patients. The problem, of course, was we could not get adequate engraftment without better conditioning, a more thorough conditioning. So in summary, the key question of the research was, is bone marrow transplant safe with CCR5 disrupted stem cells? And is it feasible? And we answered that question, yes, it is feasible. It needs better technology. The take-home message is that long-term engraftment was seen, but the level was too low. But the technology is evolving, improvement in conditioning regimen is happening. Even now, new regimens are being developed all the time, especially with antibody methods. CD117 is a very important new ingredient, and it may be the way to do transplants as outpatients that are totally non-toxic. Zinc finger disruption has improved dramatically and could be useful. And there's other methods, of course, we're using CRISPR that could be applied in this setting. So cure strategy with stem cell therapy is not yet ready, but it certainly has had a start. And I think it will be attempted again in the future with better methods. So I want to acknowledge the people that were helpful here. This is all the stem cell people, the Alpha Stem Cell Clinic, the laboratory that did the manufacturing, the clinical team under Amrita Krishnan, the funding from CERM, Sangamo, and the Alpha Clinic, and then the Sangamo staff itself that were very helpful before and during the study. And of course, the research subjects and caregivers. So I'll stop right there. Listen to questions or try to answer questions.
Great, thank you so much. That was very impressive and quite the data dump. I'm sure there are questions, clarifying questions people will have. And I'll encourage people to either put those in the chat or raise your hand. If you want to stop sharing your screen, we can go back to the Brady Bunch or Hollywood Squares view and see who has questions. But the question I had is, this obviously is very complicated and going to be very expensive and require a lot of people going through this. So where do you foresee this going so it'll be more scalable so that ordinary people might have access to that, not only here in the US, but eventually in the rest of the world where they don't have the resources that we do here?
Well, the golden fleece is direct injection vectors, and Paula alluded to that too. I think that's happening much faster than we might think. There's some really interesting stuff that's happening. If you could do direct injection with vectors that could target, let's say, hematopoietic cells or even target T cells, you may be able to really scale this up. CAR T cells, though, of course, can be scaled by making them allogeneic, and that's happening. And so from one donor, you can make 100 doses of CAR T cells, and you could imagine that would be something that could be applied. It's still going to be an expensive therapy. And how would you apply it? I tend to think in the future, a new HIV positive is going to be like a new leukemic, and you will apply extra care and treatment to that situation. And we will control HIV in other ways, but the few people that will get HIV positive and be HIV positive will then get suppressed. They'll have their load of virus suppressed with antiretrovirals for a few weeks, and then you'll give them CAR T cell therapy. Or you'll give them a vector that may modify hematopoietic cells. But my guess is that you'd use off the shelf CD4 or CD8 cells and eradicate the last cell that has tried to become latent. And at that early stage, the amount of latent virus will be much less. That's my prediction that will happen at some future time. Paula, what do you think?
That's interesting. I'm as you could tell from my talk, I'm all about let's get all these different aspects of the immune system and tweak them and improve them, and it's going to be the combination. So I'm not sure that I think CAR T cells are going to do it by themselves. Another cell that we haven't talked about is, and actually what my lab works on right now is we're engineering B cells to make, kind of fatter, fitter antibodies and use that arm of the immune system to suppress the virus as well. If I may, John, can I ask a clarifying question? In your in your cohort of patients where these CD4 T cells came back slowly, is that a consequence of the busulfan conditioning, or is it something else?
When we treat a person with AIDS lymphoma with much harsher treatment than this, we give them complete myeloablation, the CD4 cells come back faster. They will come back to normal in 9 months. We don't know why we have not seen them return faster here. The busulfan is less than myeloablative, so that shouldn't be the reason. It makes you wonder whether or not just the treatment of going through a electroporation could have played a role.
Yeah.
You know, I doubt it would be the zinc finger treatment per se, but the process of it is a tremendous amount of manipulation of the cells, much more than you'd see with a auto transplant. But some patients have taken it a long time. Some patients, even out 3 years, are just getting back to their previous state. And there was an age-related stuff too. In fact, you know, some of the donors were in their 50s, and I think it is harder at that age to recover CD4s. But if you can't recover CD4s, that is ultimately the problem. That this will never work if you can't do that. I mean, there's no reason to do CCR5 knockout if you're not going to have CD4s to enjoy the benefit. If you know what I mean.
Would adding an extra step to maybe help produce CD4 T cells rather through thymic stimulation, say growth hormone or something like that, be another step that could be added for older folks? Because if you look around this room, there's a lot of gray hair, a lot of long-term survivors with.
Yeah, only 4 of the 8 made it to the ATI threshold of T cells, and that's what they were asking. Can I take something to push my? Because they all wanted to get to the ATI.
Right.
Can I push my CD4 counts with various manipulations? And you know, we didn't know what to recommend, so we didn't recommend anything.
Yeah, it's a big gap just in treatment, right? I mean, there are a lot of long-term survivors who aren't able to get their T cells back up, and there really are no treatments out there that they've been studying. And it's such a small population. There's really not much incentive for companies to spend much money on.
Yeah, Paula, one thing you didn't mention, and I haven't heard what has happened to it, but you know, a while ago there was the use of AAV to put in an antibody. So you give a shot of AAV, and then the antibody can be produced from the transduced cell.
Yeah, people are doing that. And you know, the idea is you take these special so-called broadly neutralizing antibodies that recognize lots of different strains of HIV, and the idea is because they're so broad that even if your HIV mutates away from your natural antibodies, it will still be sensitive to these broadly neutralizing antibodies. They're like the Cadillacs of antibodies. Oh, I should say the Teslas probably. And so rather than trying to make your immune system make them, which they don't in most people, the sort of preformed broadly neutralizing antibodies are packaged in a viral vector, an AAV vector, and then they're injected into muscle typically. So you're turning your muscle cells into a factory to be pumping out these antibodies. So I think those trials are all, I mean, the monkey studies have gone quite a long way. One of the challenges is, and it's probably related to having a muscle make an antibody instead of the B cell make an antibody, as happens naturally, is that the antibodies are kind of recognized as foreign. And it's kind of paradoxically, the body makes antibodies against the antibodies. So it's like, come on, you know, it's like, what are you doing that for? So it may be that we have to produce these antibodies from the natural cell, like the B cell that makes them, to get around that problem. So again, that's what we're working on, but not as easy as injecting something into muscle and making a little factory to make the antibodies. That's for sure.
So some other questions have been coming in through the chat. So Dr. Zaia mentioned 2 strains of HIV, only one of which was permissible for the trial. Can you briefly explain what those 2 strains are and why one was acceptable and the other wasn't.
Well, remember R5 requires CCR5. The R5 strain of virus is defined as being able to get into the cells through the co-receptor, the CCR5. X4 doesn't use that. And so the problem was if X4 was present and you knocked out CCR5, it wouldn't matter because the X4 could just infect the cells anyways. So that we had to make sure that the person didn't have any virus that only had virus that could get into a cell through the R5 pathway that we were knocking out. I mean, the assumption was we would have knocked out so much that the virus can't go anyplace. Well, obviously we didn't do that, but in the design of this, the FDA required you got to make sure there's no X4 there. In fact, it's been shown that if you do a CCR5 homozygous negative transplant of a donor, like Timothy Brown had, if you have X4 virus around, that can be a deadly situation.
Mm-hmm.
So that was the reason that we could not have the CXCR4 virus.
Yeah, just to expand on that a little bit, the CCR5, and correct me if I get this wrong, is the virus that most people have that affects most cells, and CXCR4, the other one, that is the problematic one, generally only appears in people who've had, they progressed to AIDS or had a very low CD4 count, and that other virus strain, rather, variant comes up, and so you have to test it. And that's why if anybody's been on Cells Entry, you have to get what they call a tropism test, because Cells Entry is a drug that does the same thing, that blocks CCR5. And so you have to get a test to make sure you don't have that X4 virus, because as Dr. Zaia said, it wouldn't work. I hope that clarifies it. It's kind of a complicated concept of cell tropism.
I mean, one of the interesting things is we probably did affect the latency of these patients, because in the ATI, even though we only had 4, one of them went 8 months before the virus returned, and one of them went 8 weeks, which is, I realize, not very long, but it's longer than average for an ATI. The other 2 were basically the average. They were about 4 weeks until they rebounded. So that was an interesting thing. That very first ATI we did, the patient just never had virus rebound for month after month after month. And so he went farther than the 16 weeks, which we required. So, I mean, at that point, you were supposed to go back on to ART, but we got permission for him to continue. So he actually went 8 months before it bounced back.
Wow. So,
and that may be because the busulfan itself may have altered the size of the reservoir.
So somebody asked about the donors. Have you ever used long-term non-progressors or elite controllers as donors, and would that make a difference?
I think that would make a big difference. I mean, the reason the long-term non-progressors are long-term non-progressors is they have an immune system that, at least the elite, I'm really talking about elite controllers. They have an immune system that can monitor, let's call it the reservoir, and every time a virus reactivates, it kills it. And it selects for the latent virus that cannot reactivate. I mean, this is the theory now, and there's pretty good evidence that it's true. That if you really want to cure AIDS, you will have an immune system that can reproduce that and force the latent virus to be incapable of reactivating. That is, I remember that most cells that are latently infected cannot reactivate because they don't have all the pieces of the DNA there to reactivate. But the ones that can need to be killed as they reactivate. So you basically select for a latency state that is incapable of reactivation. That is the secret to a cure for HIV. So that's I think you're right. If you could get an immune system like that, you'd be in good shape.
That they call that block and lock, right? Is that same thing?
I always get those mixed up myself. I don't remember I know I've heard it before. I just don't remember it.
And then there's another question. Would these treatments work for people who have 300 or less CD4s?
That is a really good question. Ultimately, that could be the problem. I don't think that a person beginning with that low can recover without some kind of some other way to do it. You implied some kind of growth factors, thymic transplants, something like that may be necessary for that.
Yeah, no,
The car, I mean, the cars basically are that replacement. You're doing supplemental T cell therapy, let's say, to then force the virus into complete latency.
And then that's kind of a related question. What was the minimum CD4 count for this study for the inclusion criteria?
This was 450.
So that way they have room to go down and rebound.
Yeah.
Great. Other questions? Either type them in the chat or yeah, Jerry, go ahead. I'll unmute you. You need to unmute yourself, Jerry. There you go.
Could you please explain long-term survivors? I know what the words mean, but I don't understand what you're referring to. What is the science, and what are we talking about?
I was talking about the lead controller is the person who can go off of antiretroviral therapy without rebound. It's a very small number of people, but it does happen.
Well, what is the characteristic of those people? What makes them that way?
They have a T cell function that controls the virus, and it is primarily a CD8 function, but it is killing off the ability of the virus to come out of its latency. And so they were first found by treating people and then withdrawing the therapy relatively early interruption of therapy after infection was producing a small subset that could control virus without the antiretrovirals. And in analyzing those people, it seems to be an immunological function that they have. Jeff referred though to
long-term survivors are just people who've had HIV for a long time. So most of us, you know, who've been living decades with HIV, will be considered, if we're still alive, would be considered long-term survivors.
Right, but they're doing it based on antivirals.
But are doctors referring to it in terms of these people can survive without antiviral medication?
Yes. Yes.
So then there's something. Is there any research done as to what is it about their T cells that have this ability?
I can't answer that, Paula. You know, why they maintain their T cell function?
I think it's still not really understood. That it's probably going to be several different reasons. You know, it's like we put them all into one bucket. Like you hit the lottery, for some reason you've got this ability to keep the virus under control. Some of it might be genetic, some of it, you know, but people are studying these groups of people and trying to identify what's special about them, because once we know that, maybe we can think of a clever way to give that advantage and that property to everybody else. But it, as John said, it seems to be of how their CD8 T cells kill virally infected cells, and we don't really understand it at this stage.
So I typed the definitions of those two terms in the kind of a lay definition in the chat for people. But I think you were talking about long-term non-progressors. So those are people who go a long time without getting sick, but eventually have to go on meds. And elite controllers seem to be able to control the virus without ever having to go on meds. Yeah.
I'm sorry, I got mixed up between progressors and non-progressors. I didn't know we were really talking about non-progressors.
Yeah, they're non-progressors. You know, just to change the subject, as long as we have Karine Dubé here, she's very involved in working with these clinical trials that are cure-based trials, which do bring up a lot of issues. And ethical issues, number one, of course. But Karen, do you want to say anything about some of the work you're doing?
Sure. And I'm actually working on this with Jeff. So Jeff and I, we actually just got an ethics supplement a few months ago to really look into some of the ethical considerations for HIV cure-related trials. We're looking into, you may have heard of the Last Gift program. So Jeff and I are really closely involved with that. So we're looking into ethical considerations for that. And I think some of you also participated in focus groups. We just submitted 2 papers for that. We're also really interested in this ethical considerations for cell and gene therapy. We're actually working on 2 manuscripts right now for looking at potential safeguards for cell and gene therapy trials. And we're also interested in ethics of combining different regimens. So what if we were to combine 2 cell and gene therapy modality into a specific clinical trial? So Jeff and I are hoping to compile those ethical considerations, and hopefully these will guide the field. Jeff, do you want to say anything about this work?
No, I was just typed into the chat a little description of the Last Gift study, and you've heard, we've had people come talk about that before, but it's a UCSD and it's for people with HIV who are terminally ill with something other than HIV, cancer or Lou Gehrig's disease or something like that. And so what they do is they get tested, you know, before they die, they test their blood and so forth to kind of get an idea of where they are in terms of their HIV disease. And then when they die, they do a rapid autopsy within a few hours after death, so they can capture all the tissues that you couldn't get during life, you know, the brain, all the various body tissues in addition to blood, so they can get a better look. Because if you do it late, too late, the cells and the tissues degrade and they're not useful for research. So this is really innovative. They've done an amazing job of putting this together, and Corinne has done a terrific job of doing the all the work around finding out what people think about it to make sure it's done ethically and find out what people's concerns are, so we can make the study better going forward. And not just the people in the study, not just the patients donating their bodies, but also their partners, with the research team, because it's a very complicated and can be very emotional for a lot of people, because you're dealing with the end of life and death. And so it's really important to find out how this works so we can do it in a way that's sensitive and allows us to be able to keep doing the work, because it's really important for cure research. We've got to have access to that kind of information if we're going to find a cure. So thanks to Corinne and everyone who's worked on that. Because, I see another question in here. He says he has the B52 gene, which is the second class of genes found within my group of 256 participating in a long-term non-progressive study at the NIH in Washington, DC, and genes are believed to be one of the reasons. Are either of you familiar with that gene and what that means?
It's, yeah, it's an HLA.
It's an HLA, I don't know.
It means your T cells, your CD8 T cells work very well to attack cells. So again, it's everything's kind of coalescing around this idea that this CD8 T cells and their ability to kill viral infected cells is such an important component of control, natural control. And again, that's why there's so much interest in trying to engineer the CD8 T cells, as you heard from John's presentation, and also what Lentigen are doing, or enhance the ability of the CD4 T cells to help the CD8 T cells do their job. They're really kind of the most important thing, I think, at the moment. You know, we all talk about antibodies all the time and hear about that, but the CD8 T cells is really going to be critical to controlling HIV.
Great, thank you. Any other questions? You want to raise your hand or put it in the chat there. Yeah, Jerry, go ahead.
Yeah, can you hear me?
Yes, go ahead.
I have a tremendous number of CD8 cells. I've always had that as a characteristic. Does that mean that anything?
I think it means everything. I think it means that you've got the Army there. You're missing a few lieutenant generals, I think, but you've got the Army, and they're doing the best they can.
Well, I mean, just to expand on that, isn't that true for most people that the ratio of CD4 to CD8s is skewed in people with HIV? So our CD4 cells are depleted, but we have more CD8s. But as John says, we just don't have enough CD4s to kind of tell them what to do. Would that be a fair characterization?
That's a good metaphor, yeah.
Yeah. Great. Any other questions? A few more minutes before we need to move on. Well, if not, thank you so much, Drs. Cannon and Zaia. This was terrific. We really appreciate you coming to us and sharing your research. And when you have studies ready for prime time and need people, Palm Springs is just down the road from City of Hope. So
all right, good.
Please let us know. Great. Thank you both. A wonderful evening. And we'll move on to Valerio, who is now back live on camera, who can tell us about the eye care program at Borrego. So
good evening,
Valerio, take it away.
everybody. Can you hear me now?
Yes, we can hear you great.
Perfect. Actually, I want to apologize for the little technical glitch earlier, but now I'm back and I can hear everybody. Thank you, Dr. Cannon, for sharing all this information, and Dr. Zaia. It's been great to be able to learn from all of you. And actually talking about HIV, our new program at Borrego Health, who I actually manage. It's called Eye Care Program. It's a free outpatient substance abuse treatment program that helps gay and bisexual men age 50 plus, HIV positive, struggling with meth use. As we know, unfortunately, meth is present in our greater community and is also present in the HIV positive community. We started this program a few years ago. I'm very excited to be able to jump this year on it and try to do as much as we can to be able to help people who struggle with meth. Our treatment consists of 6 months, gay-specific cognitive behavioral therapy. Is offered one-on-one as well as in group settings. And we're really here to be able to help the community and help people that struggle with crystal meth. The Eye Care program is open to qualifying Borrego Health patients. And that's all I wanted to share with you for now about the Eye Care program, Jeff. But while I'm here, I also want to extend an invitation to all the providers here to our next webinar, March 25th, with Dr. Chopet. The topic of the webinar is HIV, aging, and meth use.
Great, thank you. Anything else to say about that? I put the link to the Eye Care program for Borrego there in the chat.
Thank you.
And Valerio's email.
Thank you.
And somebody asked about meth. I think he was saying meth as in methamphetamine or crystal meth.
Yeah, crystal meth, yes.
Yeah. And then someone is asking if there's a link to the webinar for the information.
I will send the link to the webinar to you, Jeff, so you can forward to the participant of this meeting. Okay. We're still working on the link.
Okay, yeah, I was looking on the website and I didn't see anything.
Yes. But we're very excited to have Dr. Chopet. Dr. Chopet is a clinical psychologist with years and years of experience working with people living with HIV and aging and methamphetamine use. So we're very excited to be able to have her as a special guest.
Yeah, Dr. Chopet has spoken many times over the years to this group.
Oh, great.
Many of us. So yeah, it's always a pleasure to hear her speak and her depth of understanding around meth and substance abuse in the the LGBT community is just amazing. So
Mhm.
we're lucky to have her. Terrific.
Yeah.
Great. Any questions for Valerio? If not, thank you so much. It's great to see you back in the desert, Valerio. As many of us remember him from his great work at Desert AIDS Project. So we're delighted to have him back and see him at his new home in Borrego. So welcome
Thank you
back.
very much. I'm very excited to be back, and I'm very, very happy to be able to work again with the community. And it's a great new chapter, and I'm very excited to be back and start this new chapter.
Terrific. Well, thank you, everyone. And thanks again to our presenters, Drs. Cannon and Zaia. They did a great job. And all that information is quite a lot to digest. So I hope everyone enjoyed it. And we will see you again next month.
Thank you.
Thank you. Have a great evening, all.
Thank you.
Bye.
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 Bye.